Welcome to our presentation. My name is Aubrey Miller, and I am an enrolling freshman at Clemson University, class of 2027. I'll be majoring in chemistry.
Hello, my name is Hannah Rowe, and I'm an upcoming freshman in the class of 2027, and I will be majoring in genetics.
Our research mentor's name was Dr. Cheryl Ingram Smith, and she works in the genetics and biochemistry department. Our research project was titled Metabolism and Cyst Formation in the Human Pathogen Entamoeba histolytica.
Our project was gene analysis of the metabolism and cyst formation in the human pathogen Entamoeba histolytica.
Important terms to know include, but are not limited to, amoebic dysentery, which is a severe form of amoebiasis associated with stomach pain, fevers, and stools. Initiation, the form and the three-layered hard-covered crust around the amoeba to protect itself from unfavorable conditions. And histolytica, which is a single-celled parasite that is transmitted to humans via contaminated water or food.
Entamoeba histolytica is a parasite that causes dysentery. According to the World Health Organization, there are about 100 million symptomatic E. histolytica infections each year, although up to 1 billion people may be infected, as 90% of the infections are asymptomatic.
Entamoeba histolytica enters the body predominantly through your mouth, which is then go through your GI system, causing infection. This is done through contaminated food and or water. The cysts, which causes infection, are spread through feces.
E. histolytica has two life forms, the growing amoeba and the dormant cyst. Cysts convert to the amoeba form in the small intestine, which then colonize within the large intestine. Here, amoeba can form cysts again in a process called encystation. Encystation requires changes in gene expression. Genes that are turned up or down at the same time may be regulated by the same proteins binding upstream at specific sequences. The purpose of our study was to look at these genes turned up or down and see if there were any similarities or points of comparison.
The method used for this research project includes the Amoeba DB program, which is an online website listed with all the amoeba genes. From there, we took the gene sequence that we were given and placed it inside the ME program, which was used to analyze the upstream regions and sets of genes that are turned up or down together to identify sequences that are shared among multiple genes and within sets. This may be a way to identify regulatory sequences responsible for initiating the encystation process.
Our results include gene EHI 056350, EHI 189190, and EHI 180130. While all of these are not identical, they are all a part of the same gene family. Further, these genes are all hypothetical proteins. EHI 134140 and EHI 120570 are nearly identical and are part of the same gene family, BSBA. EHI 184260 also contains similarities to the gene EHI 120570 and was found to be in the same BSBA gene family. All three of these are leucine-rich repeat proteins and are protein-coding genes.
EHI 157010, also known as alcohol dehydrogenase punitive, and EHI 309190, known as aldose reductase punitive, were almost identical. They were from the same gene family, which explains the results. EHI 041680, known as mucin-like protein 1, and EHI 067700, known as pathogen number 4, has different p-values. However, their motifs show homologous sequences. With further investigation, it was later discovered that they were in fact from the same gene family.
From this research, we can continue to examine aldose reductase and other gene sets with multiple identical genes to determine whether they have similar promoter structure or are differently regulated. E. histolytica BSPA gene family is a large gene family and only 3 of the genes are unregulated. Next, we can use the sequence motifs to search the genome to find what other genes belong within this family. This might give a clue as to which ones are most important for regulation. Ones that are present for tons of genes might be general promoter sequences, but ones that are less frequent might be binding sites for proteins that cause up or down regulation in response to certain signals, such as incestation.
Finally, we would like to thank the Clemson's Honor College and the EUREKA 2023 online program. We could not have this opportunity without you. In addition, we'd like to thank our mentor, Dr. Cheryl Inglesmith.
Thank you.
Thank you.